Okay. All right, people. So I'm going to just go through intermediate incomplete dominance. They're the same thing. They're just called, depending on who you are, how old you are, probably depends on what you call it. So this one says beyond Mendelian heredity, Mendel heredity. All right. So I'm just going to go through the vocab, and then I'm going to tell you about a little bit with Mendel here. But polygenic, poly meaning what, guys? Many. Genic would mean genes. Polygenic is looking at you've got more than one gene affecting a trait, determining a trait. The more genes you have, the more alleles you have, therefore the more variations you have. So we'll look at that more when we talk population genetics. Codominance, both alleles are fully expressed. I told you a little bit about calico cats, right? Okay, that's a codominance, where you've got patches of orange and patches of black. Both are expressed. There's nothing intermediate about it. So anytime you see maybe patches of stuff, like on a fur coat or something, or like stripes on flowers, usually that denotes it's a codominant. Speckled chickens, sometimes you'll see that's a codominance trait. Linked, we talked about that, two or more genes that are put together, inherited together. So Mendel, his inheritance, where he's looking at just dominant and recessive, what we've basically been talking about, is extremely rare in nature. And this is why I tell you he got lucky. You know, he picked that pea plant. Every trait that he looked at was either dominant or recessive. And so that allowed him to look at patterns, come up with different laws, like the law of independent assortment, the law of dominance, the law of segregation. But he got lucky, because that's very rare in nature with humans, animals, plants, any of that. It's very rare. Usually you see one of these four things, different alleles. This polygenetic, it can also be polygenic. Incomplete dominance or intermediate, that's what I've been talking about today. Multiple alleles, codominance. Your blood typing is a multiple allele. And one other thing. So that's why you have four different types of blood, because it's two different patterns of heredity. The multiple allele is A, B, and O. O happens to be recessive. So I named three types of blood. What's the fourth blood type? There's four types of blood. What is it? AB. That's right. So your fourth blood type is AB. That is codominant. Neither the A nor the B is completely, they're both equally dominant is what I'm trying to say. Co meaning together. So they are both equally dominant. They're showing up equal amount. O is recessive, and then you also have the Rh factor, which we don't really talk a lot about, which is known as the rhesus, named for the rhesus monkey. But that's your positive and your negative factor. That's how it was discovered. And that's another set of like proteins. So your blood type, you've got a certain amount of surface markers on your blood. And if you've got A or you've got B or AB, you've got that, like I'm A negative. So my cell surface markers are a lot of the A marker. They're just named for when they were discovered, and they just put it, and therefore, if I were to get B blood, I would not, my cells would not recognize it, and so it would get an immune response, and that was a big deal until they figured it out. So yeah, your blood type are two things. So polygenic inheritance is where you've got several genes that are affecting a trait or a character. And you've got 3 different examples. There's other examples with that. So you've got eye color, height, skin color. Hair color, same thing. This is why the more variations you've got in a trait, the more likely it's a polygenic. Let's look at height. There's at least 3 different genes. I think it's 3 genes that determine it. So if you've got 3 genes that are determining it, how many alleles are you going to have altogether? If it's 1 gene factor determining it, how many alleles do you have? You get 1 from mom, 1 from dad. 2. So if I've got 3 genes affecting it, at least how many alleles am I having? 6, that's right. So there's a lot of variation there. And this is why, this is how you know usually if it's polygenic. If you think about it, so I'm considered petite, we'll say that. I don't know. And if I go into the petite section, it goes from 4'11" over to like 5'5", I think. I'm about 5'3", not quite. Is there a span of short? Yeah, there is. If it was straight dominant or straight, in other words, if it was just 1 gene that determined it, if you were short, you'd be, let's say, 5'5". If you were considered tall, you'd be 5 or 6 foot, let's say. There would be no in between. So you either be 5'5" or 6 foot. End of story. But we don't have that. You've got variations. And so that's when you have that, you know something else is going on. Eye color, at least 3 different genes affect that. I think it's 3. Skin color, I think it's 7. Hair color, it's 5. So with your polygenic, they can either be on the same chromosome or different chromosomes. A lot of the time they're on the same one. So these genes that determine it can either be on the same or on different. The last thing I'm going to go over is that incomplete dominance. Ooh, who walked those slippers outside? So on your quiz, your last quiz next Tuesday that you're going to take, I'm going to tell you, I have to, if something is codominant or incomplete or intermediate, whatever you want to call it. I'm still going to probably call it incomplete. I have to let you know because the way you set up your Punnett squares and the how you set up the alleles are the exact same way. So I have to tell you that. But this is where it's intermediate or in between the two different traits. So like purple and white, what would be the intermediate? Yep, lavender. Think about that. I just figured this out last Friday. You got a lavender day. If you've got, if we've got one extra day in the semester, they go lavender day. So it's like a blending of the 2 days, isn't it? All right. So you've got examples. I have no idea what happened to my pictures, but they're not there now. I went all through looking for them. But
Roan cows.
yeah. And sometimes I've read it. Sometimes they consider them codominant too. So. Oh, my tea bag. I've got tea stuff. Oh well, I don't care. All right. So, you've got snapdragons with red flowers crossed with snapdragons with white flowers produces pink. It's just a blending of the two. If it was codominant, you'd have the white and the red showing up together, probably like in stripes. Have you guys seen petunias that are striped? Oh, boy.
I don't know what petunia is for
Yeah.
flowers.
It's like a little flower, like it's a,
Flower.
It's- oh my word, okay. If I can get a picture, I'll show it to you. Anyways, they love sun, so they're very easy to, you've seen them. But that would be codominant. So a lot of your plants you're gonna see that incomplete or codominance with a lot of your plants. That's why, a lot of your flowers. That's why you get so many variations. And they'll work to do that too. So, all right. So I'm gonna give you your homework. And we're gonna actually do a couple of problems together. It's not difficult, but you have to know how to do it here. Put your name on this. The first page is just information. So, when we get there, I'll tell you what you need to circle when I get everybody's out. All right. So on that front, like I said before, please put your name on it. And what you need to circle, let's just go through and circle so there's no questions on what you need to know, what you need to do for your homework. 1 and 2, circle. So flip your paper. 1 and 2, that first page is just information. So circle 1 and 2, circle 4 and 5, circle 7, and circle 9, A, B, and C. So you've got 1, 2, 4, 5, 7, 9, A, B, and C. So that's what you're going to need to do. I'm actually going to do 1 and 2 with you, so this should not be difficult. On number 4, you're going to have to figure out the genotypes before you can do your Punnett square cross, but just look how it's set up here, and you should be able to figure that out. So it says in chickens, this would be for one breed of chickens. In chickens, capital B calls for a black chicken, and a capital W calls for a white chicken. The BW combination calls for the blue chicken. I was like, I'm not trying to pronounce that. Cross a blue hen with a blue rooster. All right, so this is the exact same way that codominance would be set up. Just be aware of that. Like, you see that on the genotype, it's a capital B for a purebred, for the black chicken, capital W, purebred for a white chicken. And really, in this instance, I know on the other monohybrids, we said use homozygous dominant, heterozygous, homozygous recessive. In this instance, probably using the letters are the best bet. Why? Because both are equally dominant. You see what I'm trying to say? They are both equally. Like if you're a purebred, you're going to show up either purebred for black chicken, purebred for white chicken. The heterozygous is the incomplete, and we still use the capital letter. And you also want to notice something. In what order does it come in? Look at that BW. What's the order of it?
Blue. I mean the black.
Right, so it's alphabetical order. That's the way that we would do that. So even in your A blood typing, which I'm going to talk about next time, the order is A comes first and then the B. Same here. You go in alphabetical order. So just be aware of that. Okay. So they gave you the genotype for each one of these chickens, and they said that they're going to cross a blue and a blue. So you've got a capital B, capital W, capital B, capital W. So this is for number one that I'm doing. Then you just put your Punnett square together. Voila, done. Do you see how I did that? And I had some kids want to put the W in front because it was written first. Remember, nuance, you got to put the B in front. It's an alphabetical order. Then you've got the genotypes and phenotypes. I kind of like the way they did this. Genotypes, and then your phenotype. So Bryce, genotype, is that going to be the letters or the way it's going to look?
The way it's going to look. What do you want letters?
Right. So for genotype, it's the letters. Phenotype is the way it's going to look. So I'm going to take this. I look at each box. Capital B, 25%. You could have also written it as what? 1/4. Right. You could also do a fraction. I don't really care. Whatever is easier for you. And then I've got this capital B, capital W, and how many do I have there? 2. So I'm going to go, I always do percent. I'm going to do 50%. You could say 1/2 as well. And then the last one is WW, and that would only be what? 25%. No. That would also be 25%. So WW, 25%. Perfect. So for the phenotypes, I just have to go right across the block here. So I've got capital B, that's going to look like what? Black, and that's 25%. BW is going to look how? Blue, and that is 50. WW is white, and that is 25%. Go ahead and do number 2, and then I'll do it up here. question, anybody wanna come up and do the Punnett Square? All right, so we've got a black chicken with a blue chicken. Capital B is what you should have had. Blue is capital BW. Then I just fill in my Punnett Square box. So now I've got my genotypes and my phenotype. And I've got capital B is 50%. Capital W is 50%. And then black chickens, 50%. Blue chickens, 50%. Fini. That means done, people, or end. So I would think everybody would get this. I mean, it's not hard. So your goal is to finish out what I asked you to do. And if for some reason you are having difficulty before we look at this on Wednesday, get your little selves in here and let me look at something. All right. So set that aside. Let me just kind of stop the podcast, and then we're going to go to that mystery in Wexford. So please put that away for right now. Oops.